Life draws on the chemical elements of the universe to build viable entities. Evolution had to deal with the materials available on Earth, transforming these elements to regenerate and develop organisms. Each living being must balance an equation between its own elemental composition, nitrogen, phosphorus, oxygen, and the environment. Taste plays a crucial role here. It guides the animal towards beneficial substances and keeps it away from dangers. This sense, shaped in times of scarcity, directs us towards salt, sugar, or proteins necessary for survival. By acting as a form of sensory morality, taste combines pleasure and physiological needs. Pleasure, a biological engine, rewards the search for food and reproduction, thus ensuring the continuity of the species. When the Earth was still molten, sodium evaporated into the atmosphere. As it cooled, it solidified and fell back to the surface. The water, falling, dissolved this sodium, giving rise to salty oceans. This primitive ocean became the cradle of life. The first marine organisms had bodies whose composition reflected that of the surrounding salt water. When vertebrates began to colonize dry land, they brought with them an internal liquid rich in sodium. On Earth, fresh water was not enough to maintain this balance, hence the importance of thirst. The taste of salt, perceived by specific receptors on the tongue, instinctively guides us towards sodium. The receptors send signals associated with pleasure, prompting us to seek out and consume salt, essential for cellular metabolism. Lack of sodium leads to an irrepressible desire for salt, illustrating its vital role. Evolution has shaped our attraction to this element, essential for survival and the proper functioning of physiological mechanisms. Sodium is vital, but in excess, it shrinks cells and damages tissues. Two receptors on the tongue regulate its consumption. One encourages us to seek it out, the other slows down excess. This duality shows an evolutionary adaptation aimed at maintaining the balance of salt in the body. Nitrogen, formed from dead stars, has become an essential component of life. It constitutes amino acids, the pillars of proteins, essential to cells and the production of DNA. Although nitrogen is abundant and stable in the atmosphere, it must be transformed to become assimilable. The proteins consumed provide this nitrogen necessary for growth and reproduction. To detect these sources, animals have developed a taste receptor, umami, which rewards the consumption of foods rich in nitrogen, thus ensuring their survival and development. Kikune Ikeda discovers a unique flavor when tasting ham, cheese, and tomatoes, distinct from the four basic tastes. This perception reminds him of Japanese dashi. Back in Japan, he analyzed dashi and identified a key component, glutamic acid. In the presence of salt, this acid is transformed into glutamate, responsible for this flavor. Ikeda succeeded in isolating monozoic glutamate MSG, thus identifying a new taste category, umami. He found that natural and artificial glutamate were identical. This discovery redefines our understanding of taste, revealing the complex chemical interaction between foods and human sensory receptors. Umami, present in dishes such as bacon or bolognese sauce, results from an interaction between glutamate and nucleotides. The umami receptor, located on the taste buds, functions like a trap. It first captures glutamate, triggering the sensation of umami. The nucleotides, abundant in the muscles of animals, then bind, amplifying the signal. This mechanism produces a taste response stronger than the sum of the two parts. Discovering these interactions has helped us understand why certain food combinations are particularly satisfying, revealing the importance of nitrogen in taste reward. The umami receptor has adapted to glutamate, making the nucleotides tasteless on their own. But in the presence of glutamate, their effect is amplified, intensifying the umami taste. Chimpanzees instinctively combine raw meat and leaves to exploit this synergy. The use of tools by primates to capture ants or open oyster shells reflects a quest for new flavors. Human taste receptors detect only small molecules like amino acids, but not complex proteins. Cooking breaks down these large molecules, releasing amino acids and nucleotides, which enrich the taste of food and allow for a more intense taste perception. Our bodies regenerate by replacing almost all of these atoms each year. Getting enough nutrients is a complex task, yet it is accomplished by all living beings through taste. This sense guides animals to the necessary substances, combining pleasure and nutritional need. Taste receptors react to chemical compounds, rewarding with pleasant sensations such as sweet, salty, and umami, and warning with bitterness. Thus, taste acts as a carrot and a stick, adapted to the specific needs of each species. Living organisms obtain their energy directly or indirectly from the sun. Plants, through photosynthesis, transform light into chemical energy, producing glucose from water and carbon dioxide. Animals, by consuming these plants, recover this stored energy. 420 million years ago, plants colonized dry land, followed by worms and arthropods. These creatures then fed other species, evolving into tetrapods, our ancestors. Life is then divided between autotrophs, which draw on light, and heterotrophs, which consume other organisms. The first animals developed taste receptors, rewarding the search for carbohydrates, vital for their survival and reproduction. Ripe fruits attract animals to disperse their seeds. During maturation, they lose their acidity, become sweet and non-toxic. The sweet taste stimulates the taste receptors, activating the brain's reward system, which releases dopamine, encouraging further consumption. To save energy, some plants produce fruits with an illusory sweet taste, thanks to bracein, a protein. This molecular mimicry deceives the taste receptors of primates, except in gorillas. The latter have refined their receptors to avoid the trap. Bracein has no sweet taste for them. The taste receptors, once adapted to our ancestral needs, still reward the search for sugar and salt. Although concentrated on the tongue, they are also found in the digestive system, where they act without conscious sensations. The brain analyzes these internal signals, releasing dopamine to influence our food choices. For hundreds of millions of years, these mechanisms have guided animals to the necessary nutrients. Over time, our consciousness has added, linking perceived sensations to the body's internal signals, reinforcing beneficial behaviors. Sweet taste, once a reliable indicator of calories, triggers the release of insulin to metabolize energy. Today, sweeteners trick this mechanism. The signal no longer indicates the actual amount of sugar, thus disrupting metabolism and compromising the body's energy balance. Fungi, especially yeast, have shaped our history. Present everywhere, they transform the sugar in fruits into ethanol through fermentation. Although this wastes energy, it is a strategy to eliminate their rivals, bacteria. Ethanol, a microbicide, kills bacteria, while livestock are more resistant to it. Primates associate the smell of ethanol with a nutritional reward. 10 million years ago, the earth cooled, forests declined, and our ancestors on the ground consumed more fermented fruits. Natural selection, therefore, favored the adaptation to metabolize ethanol via the alcohol of hydrogenases. Emerging from the seas, the first land animals fed on plants, triggering a long coevolution. Plants developed physical defenses, such as thorns, and chemical defenses, producing bitter compounds to deter herbivores. These compounds, sometimes deadly like cyanide, pushed animals to develop bitter taste receptors. These receptors warn of danger before consumption. Over time, mammals have multiplied these detectors, each specialized in a group of toxins, all connected to the brain to signal bitterness, essential for their survival in the face of natural poison. Bitterness does not only signal danger. It can indicate healing properties, as with aspirin, which tastes bitter. Our taste receptors warn of these risks, because an overdose makes it a poison, but in small doses, it can be beneficial. Oleocantal, present in olive oil, is an example. This compound has shown promising anti-inflammatory effects in mouse studies. The Mediterranean diet, rich in olive oil, is associated with a reduced risk of inflammatory diseases, including cancer and Alzheimer's, thanks to these bitter compounds. Sweet taste receptors can detect even small amounts of sugar, while bitter taste receptors deter the consumption of toxins. For billions of years, these sensations have guided the food choices of animals, ensuring their survival. Taste, the yin and yang of flavors, detects molecules essential to life. Today, new flavors are being discovered, revealing keys to our evolution and opening up perspectives on our understanding of the chemistry of life.